Hello and welcome to Reprogramming of Somatic Cells to Human-Induced Pluripotent Stem Cells, or iPSCs. My name is Brian Duff. I'm Technical Lead for Scientific Support Programs here at STEMCELL Technologies. So after today's training, you will be able to describe the workflows for reprogramming different somatic cells to iPSCs using stem cell products. This is our cell culture training overview, and it really brings you through the stages of the iPSC workflow, starting with reprogramming, and then maintenance of newly reprogrammed PSCs, and then finally the stem differentiation protocols that we have to offer our customers. However, today we're going to be focusing on reprogramming and what kind of products and workflows we have to support customers who would like to reprogram somatic cells to induced pluripotent stem cells. So prior to 2006, researchers who were interested in studying pluripotency, they had to use embryonic stem cells. And embryonic stem cells are derived from the inner cell mass of the blastocyst. And in fact, many researchers had constraints at their institutes which prevented them from using embryonic stem cells. It wasn't until 2006 when Shinya Yamanaka and his colleagues developed a protocol for taking somatic cells from a patient and changing them into induced pluripotent stem cells using a range of different transcription factors, in fact, 4, known as OCT3 and 4, SOX2, KLF4, and CMYC. These became known as the Yamanaka factors. And after introducing these Yamanaka factors to somatic cells, those cells would slowly, over a matter of weeks, become reprogrammed or reverted into what are known as induced pluripotent stem cells. And now that we have this technology, it's much easier for many researchers to study pluripotency, whereas before they may not have been able to if they didn't have access to embryonic tissue. The other point to make here is that it's very critical in disease modeling. And so in disease modeling, patient cells can be taken from a patient with a specific disease phenotype, and those somatic cells from a diseased patient can be reprogrammed into induced pluripotent stem cells, which is very useful for a disease modeling researcher. And over time, many different types of somatic cells have been used successfully for reprogramming. So skin cells, blood cells, hair follicle cells, urine cells, epithelial progenitor cells, adipocytes, and other mesenchymal stem cells have all been taken from patients and reprogrammed to induce pluripotent stem cells. So really, there's a vast number of options available to your customers. So what products can STEMCELL offer? Well, we offer a range of primary cells, although it may be a little bit difficult to get primary cells to specific distributor markets. So if that's the case for you, I recommend that you speak with Mersad about the options that are related specifically to your region. RosetteSep, SepMate, and EasySep are different cell separation technologies that we can offer customers who are interested in blood reprogramming. And then also for blood reprogramming, we have our StemSpan media, which is for expansion of blood progenitor cells. Again, very important piece of the blood reprogramming workflow, which we'll go through today. Teaser E7 and ReproTeaser these are our two reprogramming media. Teaser E7 was the first reprogramming medium that we had, and it's based on the published formulation from James Thompson's lab and Gokai Chen. However, we have since optimized a reprogramming medium, and that's ReproTeaser, and that's the main reprogramming medium that we would offer to our customers. And then about 5 years ago, we launched our very own RNA self-replicating non-integrating vector called ReproRNA OKSGM, and we'll take a look at that workflow today as well. So there are a few things to consider before using iPSCs to model diseases and disorders. And the first thing to consider is the somatic cell type that you want to use. You really have to consider the epigenetic landscape of that somatic cell that your customer wants to use to change into induced pluripotent stem cells. Certain methylation patterns may restrict or inhibit the efficiency of a reprogramming. So it's something to consider. Another thing to consider is, does the somatic cell that you have access to or that you want to reprogram, does that have DNA damage? And one example of this would be a skin cell that's exposed to UV radiation or sunlight all the time. There may be more DNA damage in those type of cells compared to other cell types. And then different somatic cells have different reprogramming efficiencies. So something else to consider when you're choosing a somatic cell type. The next thing to consider would be the vector system. And we'll talk a little about this today, but there are both integrating and non-integrating vector systems, viral, non-viral integrating, viral and non-viral vector systems. And all of these have different reprogramming efficiencies as well, which have to be considered. The maintenance system. So are you hoping to take these newly reprogrammed iPS cells and put them on feeders? Or would you like a feeder-free, more reliable, more reproducible system? If it's feeder-free that your customer is after, that's definitely something that we can help with. And then taking those reprogrammed iPSCs and differentiating them into a cell type of interest. Can you do that? You have to think about, is there a possibility or is it easy for me to take these PSCs and differentiate them to the cell type of interest that I want? And are there functional assays that I can carry out with those differentiated cell types? And then finally, and very importantly, when you are thinking about modeling disease or disorders, is this system going to recapitulate the disease phenotype for you? Is it going to mimic exactly what you're trying to mimic in your model system? When you specifically think about the starting cell type, there's a few more considerations that you need to think about. The first one is cell collection. And very important, the cells that you want to collect should be accessible and with minimal invasiveness to the donor. And so what I mean there, if I give you three options, you can have blood cells, you can have urine-derived cells, or you can have fibroblasts. Which one is the least invasive, do you think? It would be urine-derived cells. You simply take a urine sample, spin it down, and then you have these urine-derived cells, which could be possibly reprogrammed. The next least invasive way would be to take blood. So taking something like blood, although you have to do a venipuncture, it's not as invasive as, for example, if you wanted to take fibroblasts from a patient, which would require a punch biopsy. You have to consider how abundant the cell type is. So if you're going after a very scarce cell type, you may have to think about how to expand those cells before reprogramming. If you're going after a more abundant cell type, you may not even have to do that. And is the cell type that you're seeking or sourcing, is it from a regenerating tissue? So if it's something like blood, of course, blood is regenerating and it's not going to have a long-term damage to the donor. So something else to consider when you're choosing a cell type to start with. I touched on this briefly just a moment ago. Is there an efficient culture system to expand and then store those cells prior to reprogramming? Something to consider when setting up your reprogramming experiments. The reprogramming efficiency of the starting cell type is very important. So typically what we see are that those more primitive cells like stem cells or progenitor cells, which are not as far along the terminally differentiated pathway as some other types of cells, they're much more easily reprogrammable. Whereas with some terminally differentiated cells, such as T cells, B cells, monocytes, they're very difficult to reprogram, or a little bit more difficult to reprogram than those progenitors. Genomic integrity is something that you have to consider. So I mentioned briefly before about external stressors such as UV light. So are the cells that you're going after, have they had less exposure to external stressors? That's something that could definitely influence reprogramming efficiency. And something else to consider is immune cells, such as B cells and T cells, they've gone through VDJ rearrangements and recombinations. And so these more terminally differentiated specialized cell types, they're a little bit more difficult to reprogram. So reprogramming efficiency with those cells can be lower. And then something that's a little bit more controversial is the downstream lineage of interest. So iPSCs, there's a thought that iPSCs are more likely to differentiate into the lineage from which they originated. And this is a concept called lineage memory. And what we mean here is that if you take fibroblast cells, which actually derive from the ectoderm, and you reprogram them to induced pluripotent stem cells, there's a thought and there's a field of thought that they are more likely to differentiate to the ectoderm than to the mesoderm and the endoderm. And likewise, if you take blood cells and reprogram blood cells to induce pluripotent stem cells, those blood cells are more likely to differentiate into cells from the mesoderm. Now, this is a controversial thought because although some people in the field believe that this is the case, other experts in the field say, no, absolutely not. Once you have a true induced pluripotent stem cell, it should have the function to differentiate into the three different germ layers, the ectoderm, the mesoderm, and the endoderm. But just something to consider if your customer is new to this area. So there's a lot on this slide, but very quickly, I want to take you through the history of the vector systems for reprogramming. So in the early days of induced pluripotent stem cells, the vectors that were used were primarily viral delivery methods. And those viral delivery methods were primarily integrative methods, insofar as the transgenes that you were introducing to the cell were actually integrated into the genome of the host cell. So using systems such as retrovirus and lentivirus, they were very efficient at reprogramming. However, you did get this integration of viral DNA into the genome, which for safety reasons is not exactly what you want to do. And as time went on, there was a trend for more and more researchers to move away from viral integrative delivery systems to non-viral, non-integrative delivery systems. And primarily now, a lot of people who are interested in reprogramming use things like synthetic mRNA, which is non-integrating and has high efficiency. And more so, people are moving towards things like self-replicating RNA, which is non-integrating, only requires a single transfection and has relatively high efficiency. And this is the example that we're going to use today when we speak about ReproRNA, our technology. So I'm going to speak now about the reprogramming of fibroblasts. And what you see on this slide are our two reprogramming media, so teaser E7 and ReproTeaser. So why do we have two reprogramming media? Well, it's actually a question of history. Teaser E7 was our first reprogramming media, and it was based on the publication, the Essential 8 publication from Gokai Chen and James Thompson. And it's basically teaser E8 without TGF beta. And it was very useful for reprogramming. So when you take TGF beta out of teaser E8, you have teaser E7, and it will actually promote, by the removal of TGF beta, you are promoting mesenchymal to epithelial transition, which is exactly what you want in reprogramming, and you're reducing fibroblast outgrowth, which is another advantage to reprogramming. So this turned out to be a very, very good reprogramming medium. However, it wasn't perfect, and we used T-Series7 and developed a proprietary reprogramming media, and that became known as ReproTeaser. So ReproTeaser has actually surpassed T-Series7 in sales. More and more people who are doing reprogramming are now using ReproTeaser. And so T-Series7 is still available for those customers who have historically used it or who want a formulation that's based on a published formulation. But newer customers tend to use ReproTeaser, and this would be our go-to recommendation for people who want to do reprogramming. So the typical reprogramming protocol for iPSCs from fibroblasts looked like this. On day 0, you would transfect your somatic cells, your fibroblasts, with episomal vectors. And then you would place those somatic cells back in their somatic cell medium, so in a high glucose DMEM serum containing medium for 3 days. Let the fibroblast recover from the transfection. On day 3, you would switch that medium out to the reprogramming medium T-Series7, and you would do daily medium changes up to about day 25 to 30. And through this reprogramming stage, you would start to see differences in your how the cells look. So the first thing is that the fibroblasts that were transfected will go through a mesenchymal epithelial transition. The cells would start to look far less striated and long and start to look much more cobblestone in morphology. A pre-iPSC colony would form, and what it would look like is a very small iPS colony. However, there would not be a defined border. And you would not pick these colonies until you started to see a defined iPS cell colony morphology. And what does that look like? Well, what you're looking for is a colony that has a defined border. The cells are very tightly packed. Under phase contrast microscopy, you're going to see a phase bright center, which is what I'm pointing out now, and that's due to multi-layering at the center of the colony. And when you start to see that, it's time to manually pick those colonies and place them into MTZR1, TZR8, or other maintenance medium. And you would then clonally expand those cell lines to establish an iPSC cell line. So that was our classical historical first reprogramming protocol for fibroblasts. However, in 2015, we launched our own vectors, so no longer using episomal vectors, now using an self-replicating RNA integration-free system. And it was called ReproRNA, and it contained the 4 Yamanaka factors, OCT4, KLF4, SOX2, CMYC, and it also contained GLIS1, and GLIS1 was added in there to enhance the reprogramming efficiency. So when you look at the vector also has these non-structural proteins, and the function of these non-structural proteins is to allow the reproRNA, once it's inside the cells, to self-replicate. That's the rationale for having these non-structural proteins there. And the vector also contains a puromycin resistance cassette. So this is used early on in the workflow to select out those cells that have been transfected with the RNA vector. And I'll talk a little bit more about that in a moment. The actual ReproRNA OK STM kit contains the vector itself, and it contains enough vector to transfect 12 wells of a 6-well plate, or 2 whole 6-well plates. The recombinant B18R protein, and I'll talk about the function of this protein in a moment. The transfection reagent and the transfection supplement, which is required. So the workflow looks like this. You would start with low passage fibroblasts at the beginning of this protocol. What do I mean by low passage fibroblasts? We recommend anything from passage 1 up to passage 5. When you go above passage 5, you will start to lose a lot of the reprogramming efficiency. So low passage fibroblasts are very important. You would grow those low passage fibroblasts in somatic cell medium. So this is a high glucose DMEM medium containing FBS. On day 0, you would transfect those fibroblast cells with ReproRNA. It's a single transfection. And then you would place those cells in growth medium. The growth medium is a serum containing advanced DMEM medium. And it also contains B18R and also, for the first few days, puramycin. So remember I said the vector has a puramycin selection cassette. So we culture the cells in a medium containing puramycin to basically rid the culture of those cells which have not taken up the vector. So you're going to get rid of a lot of background cells by adding puramycin to the medium for the first few days. B18R is in the medium because we've introduced a foreign piece of RNA to the cells. And what B18R does, it inhibits the interferon gamma response of the fibroblast cells. So it will not degrade the reproRNA vector until B18R is taken out of the medium. So that's why B18R is there. And you would culture the cells in growth medium and puramycin until day 6 and 7. Then you would switch out into repro-teaser, the reprogramming medium. No longer is puramycin needed. You have hopefully killed off all of the non-transfected fibroblasts using puramycin. However, B18R remains in the medium, and you do daily medium changes with repro-teaser every day until day 15. At day 15, the cells that have been transfected should start to have gone through mesenchymal to epithelial transition. So you start to see that cobblestoning that I spoke about before. You'll start to see really small pre-iPS colonies coming up in the well. And by day 15, there's no longer a need to keep B18R in the medium. If you're at this stage not seeing pre-iPS colonies, the reprogramming experiment was, for whatever reason, unsuccessful, and I would recommend that you go back and start again. But there's no need to continue placing B18R in the medium at this stage. There's no need for reproRNA to remain in the cells any longer. The cells that have been transfected will be well on their way to becoming iPS colonies, iPS cells. And so you can take B18R out of the medium at day 15. However, you would continue doing daily medium changes with repro-teaser until you start to see this iPS cell colony morphology of a tight border, tight defined border, tightly packed cells, and phase bright center where the cells are multi-layering at the center of the colony. And that can happen anywhere between days 20 and 28. And that's time then to go in, manually pick those colonies. And we have a video a little later on to show you how to do this. And then you take those picked colonies and you would expand them in teaser medium. And in our next lecture, we'll talk more about the maintenance media that's available to your customers. So what does the colony morphology look like? It looks very similar, indistinguishable from ES-like morphology, so embryonic stem cell-like morphology. So when you're going through the reprogramming protocol, around days 10, what you'll start to see is the mesenchymal epithelial transition. The fibroblasts that have been transfected will become less and less striated and long and more cobblestony in nature and start to come together. Around days 18, you will really start to see pre-iPS colonies. So that more round, a multi-layered morphology. However, there's no defined border at this time. And then days 25 to 30, you'll really start to see this PSC type morphology where you see a defined colony border, really tightly packed cells, multi-layering at the center of the colony. At high magnification, you may actually see some areas like this where you can see an area of differentiation on top of a fully reprogrammed colony as well. So some things to look out for when you're doing reprogramming are partially reprogrammed or transformed cells. So what you want to avoid doing is picking something that doesn't have a defined border. So you want to be sure you're not taking many contaminating fibroblasts forward. So anything like this, loosely packed cells like this, they're partially reprogrammed, or transformed or differentiated cells. So again, you don't have a defined border, but you see areas of darkness on top of a colony, or mottling like this. You want to keep away from anything like that. Probably not going to give you a good iPSC clone going forward. So I'm going to go through the technical video for ReproRNA now, and I want you to pay attention to the 3 different stages of this protocol and the important points to point out to your customers.
This video presents an easy and reliable method for reprogramming human fibroblasts into induced pluripotent stem cells using ReproRNA OKSGM. We'll be demonstrating how to use ReproTeaser neonatal human dermal fibroblasts and the ReproRNA OKSGM non-integrating vector system. The reprogramming protocol can be broken down into 3 steps: transfection, induction, and selection. The ReproRNA OKSGM self-replicating vector only requires a single transfection. For transfection, we recommend using our ReproRNA transfection reagent. Simply mix the ReproRNA OKSGM vector in a tube with the transfection reagents and incubate for 5 minutes. Take your previously plated somatic cells on a Matrigel or Vitronectin XF coated plate and replace the somatic cell medium with growth medium containing B18R. Add the ReproRNA vector and transfection reagent mix dropwise onto the cells. Gently rock the plate for even distribution. After 1 day, begin the selection process with growth medium containing puromycin and B18R. Change the medium daily for 6 days. At 7 days post-transfection, replace the medium with ReproTeaser containing B18R and continue changing the medium daily for the remainder of the induction phase. Over the next several weeks, iPS cell colonies will begin to form. When reprogramming fibroblasts, the cells will change shape, appearing more rounded as the mesenchymal to epithelial transition occurs. Around day 8 to 9, small epithelial-like cells will begin to appear. Around day 14 to 16, these cells will have developed into small clusters of tightly packed pre-iPS cell colonies. And by 3 to 4 weeks, large colonies with ES cell-like morphology will be present. In addition to fully reprogrammed iPS cell colonies, differentiated colonies and partially reprogrammed colonies may be present in your cultures. It is important to accurately distinguish between fully and partially reprogrammed iPS cell colonies to ensure successful selection and clonal expansion of the selected clones. Fully reprogrammed iPS cell colonies generated with ReproRNA and ReproTeaser will display the morphological characteristics of embryonic stem cell colonies. That is, they should have distinct borders. Cells should be tightly packed with prominent nucleoli and have a high nuclear to cytoplasmic ratio. To isolate the iPS cell colonies, either a pulled glass pipette or a 22-gauge needle is recommended. Colony isolation should be performed using a microscope in sterile conditions. Sometimes the colonies you want to select may contain differentiated cells or fibroblast overgrowth. These unwanted cell types should be removed from colonies prior to isolation. Drag the needle or pipette around the iPS cell colony to separate the fibroblasts or other unwanted cells from the undifferentiated iPS cells you are selecting. Use of ReproTeaser will generate cultures with fewer fibroblasts and differentiated cells, minimizing the need for manual removal of these cell types. To then isolate the selected iPS cell colonies, drag the needle across the colony, up and down, making a grid. Lift and collect the colony fragments with a 200 microliter micropipette and transfer to a new culture dish. The dish should be pre-coated with Matrigel or Vitronectin XF and contain M-teaser or teaser E8. iPS cell lines generated in ReproTeaser and then subcultured in M-teaser or teaser E8 typically contain very low levels of differentiated cells as early as passage 2 and are therefore ready for banking and characterization earlier than iPS cell lines generated with other methods. Successful generation of new iPS cell lines should display normal karyotype, express undifferentiated cell markers, and differentiate to all 3 germ layers using either in vitro assays or a teratoma assay. For more information about ReproRNA, ReproTeaser, and our other products for pluripotent stem cell research, please visit STEMCELL.com.
Hopefully that was a helpful video to show you the ReproRNA protocol and the important steps at each stage. So now we're going to move on to reprogramming of hematopoietic or blood cells. And we're going to specifically talk about the use of ReproTeaser here now. This is a very useful decision tree that can be used to help guide customers on the correct workflow for reprogramming blood-derived cells. So at the top of the tree, it's really important to ask the customer if VDJ rearrangements are either acceptable or not acceptable. If they are acceptable, it's fine for the customers to go ahead and reprogram using PBMCs or TMB cells. And really, they won't be limited in their decisions after that. And they will be able to reprogram their PBMCs or T and B cells to iPSCs. However, we don't necessarily support this workflow. What we do is we support a workflow for reprogramming of progenitor cells. You'll have much higher reprogramming efficiency using more naive cell types such as progenitors. So if progenitors are the way your customer wants to go for blood reprogramming, the next important question to ask them is about their sample size. Do they have access to a large amount of blood or access to a small amount of blood? What do I mean by large or small? If they have access to a large amount of blood, typically that means greater than 50 mils. One of the best options for them is CD34 reprogramming. So taking those very few CD34 cells that are available in a large sample of blood, so greater than 50 mils, and reprogramming those cells to iPSC cells. Or if they only have access to a small sample, so anything less than 50 mils of blood, really what they want to do there is go after progenitors that are a little more plentiful than CD34 cells. And in our experience, that's erythroid progenitor cells. And take those progenitor cells and expand them and then reprogram them to iPSCs. So here's the workflow for taking peripheral blood CD34 cells and reprogramming them. So like I said, if your customer is interested in this workflow, they really need to be starting with minimum 50 mils of peripheral blood. Even with 50 mils of peripheral blood, the number of CD34 cells that they will isolate will be so small, they really will need to expand those CD34 cells before reprogramming. So the options here is they can start with cord blood or peripheral blood. CD34s are more plentiful in cord blood. However, cord blood volume is often a lot smaller than peripheral blood. We would then get our mononuclear cells by using a setmate 50 tube and Ficol to separate those mononuclear cells from granulocytes and RBCs. And then we take those mononuclear cells and we put them through a positive selection using our EasySep immunomagnetic cell separation. And we are positively selecting the CD34 cells there. So those CD34 positive cells that we get after this, there's still not enough of them to do a reprogramming experiment with. So what we recommend is that you should expand those CD34 cells using our STEMSpan STEM2 medium with our CD34 expansion supplement for 7 days. This will allow the overall number of CD34 positive cells to increase in your culture, and then you will have enough to transfect for the reprogramming experiment. So once expansion for 7 days is complete, you would then transfect the cells using episomal vectors. So stem cell technologies do not carry episomal vectors. We recommend using Epi5 vectors from Thermo Fisher for this portion. ReproRNA OKSGM will not work on blood cells, so it's very important for blood cells that you follow the episomal vector reprogramming protocol. And then after transfection with episomal vectors, we then place those CD34 transfected cells back into STEMSpan SFEM2 and expansion supplement for 3 more days. So we put them back into the medium that they've come from. They've gone through a very stressful transfection, so we don't want to stress them anymore by placing them in a different medium. After 3 days, we do a fed batch change into ReproTeaser, which I'll show you on the next slide. And you keep those cells in ReproTeaser for 18 to 25 days until you see this new putative human iPSC colony coming up. And then again, like you saw in the video, you would pick that iPSC colony into mTeaser 1, teaser E8, or any of our maintenance media. Okay, so what exactly does the blood reprogramming workflow look like? Well, like I said, you would take those cells on day 0, transfect them with episomal vectors. This is after they have been expanded for 7 days in STEMSpan medium. You would place them back into STEMSpan medium with the appropriate supplement. Keep them there for 3 days. On day 3, you would then add 1 ml of ReproTeaser medium without taking away any STEMSpan medium from the well. On day 5, you would add another ml of ReproTeaser, again, without taking anything else out of the well. So at this stage, the well is very full, but it's very important not to do a medium change until you reach day 7. The reason for that is these hematopoietic cells will be in suspension after they've been transfected, and it may take them some time to sit down and attach to the culture wear, to the coated culture wear. So to ensure that you are getting as many reprogrammed colonies growing as possible, it's important not to remove them from the culture too soon. And so you give the cells every opportunity to sit down to attach to the matrigel vitronectin coated plate. So by day 7, anything that has been transfected should definitely be sitting down at this stage. On day 7, you can do a full medium change into the reprogramming medium ReproTeaser. And you continue doing daily medium changes, you'll start to see the cells cobblestoning as you saw with the fibroblasts. However, with blood reprogramming, you'll hopefully see a lot less background. And then anywhere from day 21 to 28, you'll see this putative human iPSC colony coming up where you have your defined border, your prominent nucleoli, high nucleus to cytoplasm ratio at high magnification, and that dense center due to the multi-layering at the center of the colony. That's when it's time to pick that colony into our maintenance media for clonally expanding that line. So when you think of the workflows that we have for reprogramming, we can support reprogramming of human fibroblasts, CD34 cells, erythroid progenitors. People can also reprogram PBMCs, although it's a little more heterogeneous. There's a little bit more heterogeneity here because you have different cell types. And we can offer medium to expand the somatic cells to start off with, products to reprogram the cells, medium to culture reprogrammed cells, and then obviously medium to culture iPSCs. So for human fibroblasts, we recommend a high glucose DMEN, 10% FBS, and non-essential amino acids. The vector we recommend is now ReproRNA OKSGM. Historically, we recommended episomal vectors, but now we have ReproRNA. We will offer this for fibroblast reprogramming. The reprogramming medium that we recommend is ReproTeaser. Previously, we recommended Teaser E7. And historically, if customers have wanted a published formulation, they can use Teaser E7, but ReproRNA is our first option. And then once you have your putative iPSC colonies, you would pick them into any of our maintenance media. So mTeaser1, Teaser E8, or mTeaser Plus. And we'll talk a little bit about those media in our next lecture. For CD34 cells from peripheral blood, we would expand those cells in STEMSpan media plus CD34 expansion supplement. Remember, for blood reprogramming, ReproRNA does not work, so we would recommend episomal vectors. Our reprogramming media of choice here would be ReproTeaser, and of course, the same medium to culture the iPSCs once you have them. For erythroid progenitors, we would recommend STEMSpan SFEM2 medium plus the erythroid expansion supplement, episomal vectors again, and ReproTeaser medium for reprogramming, and again the same maintenance medium to culture the newly reprogrammed cell line. And finally, if people want to start with PBMCs, what we would recommend is expanding them in the STEMSpan and erythroid expansion supplement first. Again, episomal vectors, ReproTeaser, and then the same maintenance media after the reprogramming is complete. So here's an overall workflow of the PSC continuum. And what we spent a lot of time on today was the reprogramming section. And we spoke about the different cell separation products that we can offer our customers who are interested in blood reprogramming. Teaser E7 was our historical reprogramming medium. However, more and more people are shifting to ReproTeaser now. And for a vector for reprogramming fibroblasts, ReproRNA is the way to go. We can also offer small molecules and cytokines should customers request them for reprogramming also. I hope that was very helpful today. Thank you for your attention.